Good morning, students. This is Murthy, sir. In the last class, we discussed about human reproductive system. Today, we will learn about in vitro fertilization and cloning techniques. In vitro fertilization is popularly called IVF. It is an advanced technique in medicine. Students, in vitro fertilization technique, the egg of the female is fertilized outside the body. Fertilization of the oocytes, it takes place in the laboratory. This is in contrast with the natural fertilization which occurs in the female body in the fallopian tubes. Students, in case of some women, if the oviducts are blocked, they will not be able to give birth to children because sperms cannot reach the eggs for fertilization. In such cases, doctors, they collect freshly released egg and sperms and keep them together for few hours for IVF. In case fertilization occurs, the zygote is allowed to develop for about a week and then it is placed in the mother's uterus or mother's womb. Students, in IVF technique, only fertilization is allowed to happen in laboratory conditions. The complete development of the baby takes place in mother's uterus or mother's womb. Students, in this photograph, you can see the first test tube baby Louis Brown and her mother. Students, babies born through IVF technique are called test tube babies. Students, the first successful birth of a test tube baby, Louis Brown, it occurred in the year 1978. Robert Z. Edwards, the physiologist who developed the treatment, was awarded the Nobel Prize in Physiology or Medicine in the year 2010. Subhash Mukhopadhyay was a physician from Kolkata, India, who created the world's second and India's first child using IVF technique. The name of the baby is Durga. Students, South India's first test tube baby is Kamalaratnam. Students, here you can see the great scientist who developed this technique, Robert G. Edwards, who was awarded Nobel Prize in the year 2010. The scientist who developed in vitro fertilization. Students, this is the photograph of the oldest woman who gave birth to a baby through IVF technique at the age of 66 in the year 2004. Her name is Adriana. Students, now we will move on to another interesting technique, modern technique in biological sciences. It is called cloning. Students, cloning is the production of an exact copy of a cell, any other living part, or a complete organism. Students, cloning is the process of making a clone, a genetically identical copy of an organism by placing the nucleus of an unfertilized ovum with the nucleus of a body cell from the organism. Students, in the year 1996, cloning was revolutionized when Ian Wilmot and his colleagues at the Roslin Institute in Edinburgh, Scotland, successfully cloned a sheep named Dolly. Dolly was the first cloned mammal. Students, you know, mammals are highly evolved in animal kingdom. Here you can see the photograph of Ian Wilmot and Dolly, the first cloned mammal. Students, Dolly was born on 5th July 1996. Students, now let us learn about the actual process. Ian Wilmot and his colleagues transplanted a nucleus from a mammary gland cell of a fin-dorsed sheep into the enucleated egg of a Scottish black-faced Eve. The nucleus-egg combination was stimulated with electricity to fuse the two and to stimulate cell division. The new cell divided and was placed in the uterus of a black-faced Eve to develop Dolly. Dolly was born months later. So students, this is indeed a great achievement in the field of biological sciences. Here you can see the diagrammatic representation of the process of cloning, how it takes place. You can see here. Students, Dolly was a healthy clone of the Finn Dorset sheep and produced several offsprings of her own. Offsprings means her own babies. Students, unfortunately, Dolly expired on 14 February 2003 due to lung infection. Students, creating a clone of a mammal is a great achievement in the field of biological sciences.